Dr. Schultz and I are very honored to be here at SAWC giving a presentation on proactive biofilm-based wound management. Dr. Schultz and I have given presentations before across the country, and it has become abundantly clear that he's the science guy, I'm the clinical guy. To go into his credentials, he is a professor of the Department of Obstetrics and Gynecology and the director of the Institute of Wound Research at the University of Florida in Gainesville. And I am the Chief Executive Officer of MedCentris, which is a multi-specialty wound healing group, as well as limb salvage group, as well as a management group based out of the New Orleans area. And I'm the director of the Baton Rouge General Center for Advanced Wound Hyperbaric Medicine in Baton Rouge, Louisiana. Our disclosures are listed in your packet. Dr. Schultz and I always strive to make sure that every presentation we give is evidence-based, and that will remain so today. So our learning objectives for today, after we're finished, we hope you all have an understanding of how to examine a proactive biofilm-based wound management strategy aimed at reducing and preventing biofilm reformation, how to employ a comprehensive and proactive BBWM, that's the phrase we may be using today, BBWM, biofilm-based wound management protocol, including sharp debridement, application of broad-spectrum antimicrobial therapy, followed by adjunctive therapy, be able to review components of an ideal antimicrobial technology, and explain the science of polyhexylene biguanide hydrochloride, otherwise known as PHMB. So we will no longer be using these long terms. We'll shortcut it. PHMB will be what we use for polyhexonide throughout the presentation. Our topics, we're going to go over how biofilm impedes healing of chronic wounds, how biofilm is common but difficult to diagnose, why biofilm is hard to eradicate, how aggressive debridement is essential, but biofilm can reform rapidly after aggressive debridement, the limitations of antimicrobial therapy of biofilm, the ins and outs of PHMB, how it's an excellent topical antimicrobial, the importance of native collagen matrix in wound healing, PHMB within a native collagen matrix, and we actually have a case study to show you a little bit of the strategy on how to address biofilm, how that can be approached. So with that being said, I'm going to turn it over to Dr. Schultz, and he's going to go over the science, and then I'll get to the clinical after he's done.
What I'm going to do is review some of the important fundamentals, as Dr. Carpenter indicated, and then he's going to focus in on a really interesting and very important way to take this basic microbiology and biochemistry stuff that I'm going to give you and then translate that into something that is clinically useful for you as wound care providers. So let's just start very quickly with definitions. What is a biofilm? Well, biofilms are colonies of bacteria that are encased in EPS, and this is another one of those acronyms that biochemists love to use. That's exopolymeric matrix or exopolymeric substance. This is biofilms are due to free-floating microbes, which are those single planktonic bacteria that we all grew in microbiology class and seeded out and grew colonies on agar plates. But instead of those free-floating, actively proliferating, non-attached planktonic bacteria, they will convert over into firmly attached or sessile bacteria. And they do that by switching their patterns of gene expression from a gene pattern that is predominantly directing the bacteria to take most of its energy and proliferate as rapidly as it can into a phenotype that is taking most of its energy and frankly synthesizing this exopolymeric substance that is encasing them and protecting them and helping them attach to surfaces. And they do that by switching their patterns of gene expression under a system that's called quorum sensing. And basically, what that means is when bacteria are single free-floating planktonic bacteria, they're synthesizing these unique signaling molecules called quorum molecules. And there are three major chemical classes of those based on gram-positive or gram-negative or a common phenotype of the quorum molecule. When the density of the bacteria gets high enough, when they reach a quorum, the concentration of the quorum molecules becomes high enough, the quorum receptors on the bacteria surface sense that, and it changes their pattern of gene expression. So if you will, this is kind of like a hormone sensing system between bacteria. The reason bacteria evolved this capacity to do quorum sensing and shift from planktonic to biofilm phenotype wasn't to cause chronic wounds in us. It was to protect themselves from all of their natural predators out in the environment, mainly from the single-cell big amoebas that want to encase and eat the bacteria, or the bacterioviruses, the bacteriophages that want to infect the bacteria, proliferate, and rupture and kill the bacteria. So it's just accidental that the formation of the biofilm phenotype that's protecting the bacteria out in their normal environment in the rivers and the streams and the oceans actually accidentally causes chronic inflammation in us, because our amoebas, our neutrophils and macrophages, can engulf and kill the bacteria effectively when they're in biofilms. And so they're very difficult to eradicate. And as I'll show you, and as Dr. Carpenter will show you, it really becomes a key factor that directly impairs wound healing. The chronic inflammation caused by the biofilm phenotype that's difficult for our immune system to kill really causes elevated levels of protease and reactive oxygen species that makes the presence of the biofilm detrimental to healing. So in the beginning, what is a bioburden? Well, let's make sure we're using the term as we all understand it. It is defined as the number of bacteria living on a surface that's not been sterilized. In other words, really, the bioburden are all of the viable bacteria, both in the planktonic, which are the free-floating, as well as the attached or the biofilm bacteria. And even the free-floating bacteria are not totally independent. As I indicated, they have these quorum sensing systems that help them get information about what is in their surrounding environment. So we have the whole phrase of bioburden. Every bacteria, regardless of whether it's biofilm or planktonic, that's there. So we also have this spectrum of the bioburden. At the lowest end of that spectrum of increasing severity of the bioburden is contamination. This is the presence of non-replicating microorganisms on the wound surface, and they basically do not cause a significant problem clinically. Now, essentially all open wounds are contaminated, but the bacteria counts are low enough, and in almost every case initially the bacteria are in a planktonic phenotype. So our immune systems do a very good job of keeping these contaminating bacteria from exploding into a pure, true acute infection. And wound healing can occur even in wounds that are contaminated. Now, in generality, multiple studies have shown that when the total planktonic bioburden is about 10 to the 2 to 10 to the 3 colony-forming units of most bacteria, there are some very virulent exceptions, as I said, our immune system can normally handle that. Our antibodies and our phagocytic cells can normally handle that. Well, going one step up, this bioburden spectrum is the state of colonization. And these bacteria have begun to adhere onto the superficial tissue. That's this sessile bacterial state. They're not completely free-floating. And they have begun to form colonies without generating a huge immune response. In other words, these bacteria are there. They're slowly starting to proliferate, but again, our immune systems normally can handle these well enough, and typically they're not associated with a significant delay in healing. So this is a visual representation of this bioburden spectrum, especially emphasizing the concepts of biofilm formation. So down here on the bottom, this is our bioburden spectrum of contamination, colonization, and then, as we'll see, this concept that evolved called critical colonization, in which there's some function, some aspect of the bioburden that's impairing healing. And then at the full end, the highest end of the spectrum is the true acute spreading infection. That can even be sepsis that's life-threatening. But what we now appreciate is that in this area of critical colonization, we didn't quite understand what was critical about it 20 years ago when this term was being evolved. But now we appreciate the reason it's critical is because that's when the biofilm is starting to form and causing the chronic inflammation that's going to impair healing. And so again, this is kind of what we need to think about in terms of what's happening to the bacteria as they become attached, planktonic, proliferating, quorum sensing, and eventually become attached and begin to convert mainly into the biofilm phenotype. So here's this term again, critical colonization. And this means that the wound is not able to maintain a real balance between the bioburden, the total form of the bacteria, and an effective immune system. In other words, saying that differently, as these bacteria begin to attach and convert to biofilm protective phenotype, our immune system now isn't effective at engulfing and killing them, and our antibodies are not able to really interact effectively with them to be optimized so that they can also be killed by our complement system. And there are multiple papers now that clearly associate the critical colonization state, this initial conversion into the biofilm, with the delay in healing. But as I was saying earlier, initially 20 years ago, we didn't know or didn't appreciate the concept of biofilm, so we just said it was critical. We weren't quite sure why it was critical, but now we're much more confident that it's mainly due to the biofilm. Now, the other thing is that it is not a pure clinical infection. Now, you as clinicians understand the difference between the signs and symptoms of infection and the signs and symptoms of inflammation. So bacterial biofilms typically do not cause the classic signs of infection. They do always cause the clinical signs and symptoms of chronic inflammation. So that's important for us to understand that. Now, as we look through the normal phases of wound healing, we know that first there's hemostasis, and then as the clot forms and the platelets degranulate, our inflammatory cells come in, because those platelet contents have chemotactic factors for our neutrophils and macrophages. So that moves the wound into this inflammatory phase. And if there isn't a conversion of the bacteria into an acute infection or into this biofilm phenotype, normally the inflammation phase will peak at about 5 days and about 10 days. Most of the time, the inflammation signs will go away and the wound will move into a clearly active repair phase. But what we've found, and what many laboratories around the world now have found, is that instead of moving out of this inflammatory phase, the wounds that fail to progress become chronic because they get stuck in a chronic inflammatory phase. And again, what we're now appreciating is that it's most cases, the reason they get stuck in that chronic inflammatory phase is because the planktonic bacteria have converted to a large extent over into the biofilm phenotype. And there are data that I'll show you that is unequivocally establishing that the formation of the biofilm, the protected structures that the bacteria are in, actually impairs healing due to the chronic inflammation elevating the level of proteases. We can't, biochemists love flow charts and enzyme sequence pathways. Well, this is an example of trying to generate a biochemist view of how acute wounds convert into chronic wounds. And basically what happens is when we have an acute injury, if our immune system is not able to completely prevent the critical colonization, then what will happen is that it will shift the wound into this chronic inflammatory phase. And we can measure that by noting that there are lots of pro-inflammatory cytokines if we sample wound fluids or wound biopsies of chronic wounds compared to acute wounds that are healing. The levels of tumor necrosis factor alpha, IL-1, 6, etc. are going to be 50 times higher than what we see in a healing wound. And those pro-inflammatory cytokines do exactly what they normally do. They draw in more and more neutrophils and macrophages, and those neutrophils and macrophages activate. They secrete the proteases and the reactive oxygen species that they normally use to remove the damaged matrix from an acute wound and also to engulf and kill the bacteria through the generation of reactive oxygen. But what happens is when the bacteria are in that protected biofilm phenotype, our immune cells can engulf and kill them, and our antibodies can't penetrate it. So now the levels of inflammation and the levels of proteases remain very elevated for long periods of time. In fact, if we measure the level of proteases in chronic wounds, they average 50 times higher than an acute wound, and in many of the worst cases, they're over 250 times higher than what we see in a healing wound. And they are not totally selective in removing just damaged matrix. In fact, they have what biochemists call off-target effects. They begin to destroy growth factors and receptors in the new functional matrix that's being deposited by the fibroblasts and the epithelial cells. That results in decreased cell proliferation and migration, which you see clinically as an acute wound that's converted into a stalled or chronic wound. So normally what happens is you have to break this amplifying inflammation cycle optimally by removing the source of the inflammation, which in most of these chronic wounds now we know is becoming due to the biofilm. So there is this continuum of the bioburden. So remember, we talked about contamination, colonization. We talked about critical colonization and true spreading and systemic infection. This is where the biofilms are having their major impact in this area previously we called critical colonization. And so your challenge as clinicians is to recognize when a biofilm is likely to be present in a wound, and then how to implement effective biofilm-based wound care, which is what Dr. Carpenter is going to tell you about. So there was a panel of experts, both clinicians as well as basic scientists, microbiologists, and this panel came together, and Dr. Carpenter was the lead author on this, to try to put together a consensus recommendation of how to recognize, diagnose, recognize, and treat biofilms. And some of the key findings from that panel were that the significance of a biofilm is often not well appreciated. I think the field is becoming more and more educated about this, but I think still there are lots of people that don't really fully appreciate what a biofilm is and how it impacts healing. And even the panel felt that even when the wound looks pretty clean, biofilm is frequently present and delays healing. In other words, it's not always the sloughy, infected-looking wound that is the only wound that's going to have a biofilm. In fact, 80% or more of the chronic wounds that don't look like they're clinically infected when we take samples and analyze carefully for biofilms are present. And again, the panel felt that there was unequivocal evidence linking the presence of a bioburden with a high component of biofilms that actually is critical to impairing wound healing, and that's through this formation of the high proteases. And that the proteolytic enzymes generated by the biofilm can frequently degrade collagen dressings when the bioburden is not adequately controlled. And Dr. Carpenter will go into this more about how to do good step down, then if necessary step up therapy to remove the biofilm, get the wound out of an inflammatory phase, and then use advanced treatments to finally begin to improve healing. And there's obviously a need to educate, which is kind of what we're doing here now. So another concept that the panel felt was important is that biofilm is common but difficult to diagnose. And back again to that bioburden continuum, essentially all chronic wounds after they've been there for 4 weeks or more are contaminated, and they always have low levels of planktonic bacteria. But those levels of planktonic bacteria, remember our immune systems, unless it's in an immune-compromised patient, frequently are not the major cause of why the wounds are not healing. And when planktonic bacteria convert into biofilm, the wound progress is stalled, and therefore the bioburden control becomes critical. Really, this biofilm-based wound care concept becomes critical. And now there are even more recent meta-analysis and publications that show Initially, we thought it was about 60%. Now it's closer to 90% of chronic wounds contain biofilm that we can identify with rigorous methods. Biofilm diagnosis is difficult. The presence of biofilm can be underdiagnosed, and Dr. Carpenter and I were talking about this this morning. And that is the fact that when you take a swab or a curettage or a biopsy and you send it over to your really good clinical microbiology laboratory, they will give you good data only on planktonic bacteria. They do not process the samples to ultrasonically disperse the biofilm out of this protective, relatively dormant growth state into a form where the bacteria will actively grow on typical agar plates. So it doesn't mean that your clinical microbiology lab results are not useful. They are useful, but they only tell you about the planktonic culturable level of bacteria that's present and what that laboratory is capable of doing. Wound cultures may point to antibiotic treatment, but it does not indicate the level of biofilms, and especially it does not indicate necessarily what's going to be effective to kill them. So again, from the expert panel discussion and recommendations, wound biofilm is extremely common. I think I've heard my clinical colleagues say if you have a chronic wound that's been there for 6 weeks, assume 100% of the time there's a biofilm component somewhere within that wound bed. Wound cultures only detect the planktonic bacteria. They do not detect or identify biofilm. And all wounds should be treated on the assumption that a biofilm is already present or is highly likely to develop unless you do biofilm-based wound care and interventions, which Dr. Carpenter is going to talk to you about. Now, another critical thing for us to understand and what the panel dealt with was that biofilms are difficult to kill. Again, bacteria didn't evolve the capacity to make biofilms 2.3 billion years ago in their evolution. They evolved that capability to avoid being eaten and killed by amoebas. But this exopolymeric matrix substance is the bacteria's way of protecting themselves. And so there are within a mature biofilm community the presence not only of this exopolymeric substance, but there are also persister bacteria. And this is, again, an extremely important concept for all of us to understand. As I'll show you, if you treat planktonic bacteria with antibiotics, it kills them really well. That's because they're rapidly metabolizing, and antibiotics only kill metabolically active bacteria. They kill the bacteria by interfering with bacterial enzyme systems and transport systems. If the bacteria are metabolically dormant, that is, not using all those enzyme systems, the antibiotics have almost no effect because inhibiting something that isn't active doesn't make a big difference in the survival of the bug. So persister bacteria are a major factor in why mature biofilms are not effectively killed by the antibiotics that are very effective at killing planktonic bacteria. In addition, within a mature biofilm community, the metabolically active bacteria are on just the surface of that structure, and so they consume the oxygen, and that creates a reduced level of oxygen in the center of these biofilm masses, and that's frankly where strict anaerobic bacteria can survive. So Dr. Carpenter and I were talking this morning again about DNA identification of bacteria, and the fact that when you do DNA identification in chronic diabetic foot ulcers or pressure ulcers, about 60% of all the bacteria species that we can identify by DNA sequencing are strict anaerobic bacteria. And a large number of those are associated with the areas of biofilms that have low levels of oxygen. And then finally, all of the typical chronic wound biofilm communities are polymicrobial. There isn't just one bacteria there. And the sum of all fears is when you have a polymicrobial biofilm community and you have a sensitive Staph aureus here, and right beside them is a cluster of methicillin-resistant Staph aureus secreting all those penicillin-binding proteins and beta-lactamases that give them a genetic resistance to the antibiotic, and the bystander effect protects the sensitive bacteria that are right beside them because the diffusion provides an umbrella, a zone of protection, a bystander effect. So the concept of being able to have synergistic bacteria interactions and allowing sensitive bacteria to survive when they normally wouldn't is another reason that biofilms are a problem. So the panel realized that it's important to try to understand how the biofilm is difficult to eradicate, and especially to understand how that biofilm can disrupt the normal molecular physiology within healing wounds. And this diagram just shows that, which really is indicating that eventually what happens is when the biofilm forms and the chronic inflammation occurs and we have the release of the proteolytic enzymes and the reactive oxygen species by neutrophils and macrophages trying to eradicate and kill the biofilm, that we have those bystander effects. So again, from the expert panel discussion and recommendations, the key concepts were that biofilm-associated bacteria can be extremely difficult to kill. Antibiotics are effective only against metabolically active bacteria, and biofilm-associated bacteria have a component usually within the center of the biofilm that are metabolically inactive, the so-called persister or dormant cells. And that both resistance, that is true genetic, inheritable, permanent resistance, as well as tolerance, can cause antimicrobials to be not effective against biofilms. So how do we start with biofilm-based wound care? Well, the real important aspect to understand is that if you can't kill the bioburden and the biofilm, the best thing you can start with is debridement. So critical within the initial stages of biofilm-based wound care is an effective debridement. You can do that by multiple ways. You understand better than I that there are certain patients you can't do sharp debridement on. So you're going to have to adapt the most effective and most rapid debridement technique you can for the patient. And in addition, biofilm-based wound care emphasizes that you can't just debride the wound and assume that the biofilm is not going to come back, because data that we've published with Randy Walcott shows that in patients that are effectively debrided, if they're treated with a non-microbicidal barrier dressing, that's the FDA term for all those antimicrobial dressings you use, then within 3 days, the biofilm in those patients will have reformed. And that's because most of the things like wet to dry gauze dressings allow bacteria to penetrate right through. They contaminate, colonize, and they convert into biofilms, and it's right back where it was before. So again, aggressive debridement is essential, but biofilms will rapidly reform, and particularly you need to understand that you need to combine debridement with an effective microbicidal treatment to prevent the biofilms from reforming within about 3 days. The panel's recommendations and discussions also indicated that successful wound management requires successful biofilm management if you're going to heal the wound. You need to use this step-down approach, which means start with the most effective treatments first. Don't wait for another 4 weeks to see if that dressing you're going to put on there has any effect. Assume you have a biofilm, do effective debridement, use your most effective antimicrobial and anti-biofilm dressing that you can to step down to a state where the inflammation is under control, the proteases are under control, because you control the biofilm and the planktonics. Then if you need to add treatments to the wound bed to jumpstart, to promote a more aggressive healing environment, not just an antimicrobial, but a healing environment, then you can step up your treatment to some of the advanced wound care treatments. And again, Dr. Carpenter is going to talk to you about that. And I'm just going to show you very, very quickly the limitations of topical antibiotics. Antibiotics kill planktonics really well, but they're not going to be able to kill the bacteria that are deep within the biofilm because of the dormant biofilm and because of the difficulty in penetration through this exopolymeric matrix substance. So it will shield and hold the bacteria community, and this provides an effective tolerance to most of the things that we use to try to kill the planktonic bacteria. This diagram basically is conveying the concept that When you have a wound bed, you're going to have a lot of material initially on the surface. A lot of that is fibrinous slough. It's not biofilm. It's the result of the inflammation caused by planktonic and biofilm bacteria. Those pro-inflammatory cytokines open the tight junctions in the capillaries, and out comes a large amount of plasma-rich protein. As I was indicating earlier, just to show you how tolerant these bacteria become in the presence of a biofilm phenotype, if we grow Pseudomonas aeruginosa as a planktonic culture and expose it to 50 times the MIC for tobramycin, within about 4 to 5 hours, all of the planktonic bacteria are killed. That's what antibiotics do well. They kill rapidly proliferating, rapidly metabolizing cells. On the other hand, if we take those same sensitive bacteria and grow them in a mature biofilm phenotype and treat them for 4 days with that same level of antibiotic that killed every planktonic bacteria in 4 hours, after 4 days it's killed 1 log. Now if we take these bacteria that are in the biofilm and are tolerant to the antibiotic that was killing the planktonic, and we ultrasonically disperse them back into single planktonic bacteria and expose them to tobramycin again, kills every one of them. So this is not genetic permanent resistance. That's a different concept than transient tolerance. The biofilm provides tolerance because of the physical environment that the bacteria are in. Antibiotic resistance is a permanent genetic alteration in the bacteria, and it doesn't matter what conditions they're grown under, the antibiotic isn't going to kill them. But just to reemphasize, this shows you the enormous tolerance that bacteria that are sensitive to an antibiotic in the planktonic state develop tolerance when they're in the protected biofilm. This is again an indication of why systemic antibiotics have limited effects on biofilms. I showed you that, and it's just to give you another clinical example. When a patient has osteomyelitis or an infected orthopedic hardware, that's a biofilm-based disease, unequivocally. Why do they have to be put on 2 months of oral antibiotics? Because the bacteria are in a biofilm. You've killed every planktonic bacteria in that patient's area of the osteomyelitis within 24 hours. That 2 months of antibiotics is slowly killing some, and then finally most of the bacteria that's in the biofilm by killing that outer shell of metabolically active bacteria. So it's almost like peeling an onion. And it takes weeks to kill the dormant persister bacteria as they slowly reactivate as the outer layers are killed. So again, topical antimicrobials can work wonderfully well against planktonic bacteria. There's a spectrum of response to different kinds of topical antimicrobials. And you have to balance killing the biofilm with not killing every wound cell. And so, again, as Dr. Carpenter is going to indicate, PHMB is one of those antimicrobials that has a good enough cytotoxicity profile and a good enough biofilm killing profile that it is a very effective and a good choice for using to try to limit and kill the biofilms. So finally, the expert panel recommendation was that topical antimicrobials need to be better understood and appreciated by what they can do and what they don't do well. And it's also important to be aware that some antimicrobials have such toxic effects on the wound cells that their use has to be very cautiously and very carefully used and monitored, because if you kill too many of the wound cells, even if you've killed the biofilm, you're still going to impair healing. So I'm going to stop there, and Dr. Carpenter is going to come up and talk to you in more depth about this PHMB antimicrobial and especially how to do biofilm-based wound care clinically. Shaun.
Okay, so you all have just heard a very in-depth scientific explanation of what is biofilm, how does it form, why is it hard to eradicate. So when you go back to your clinics and you say, I just went to SAWC in San Diego, and you explain to your patients this science and their eyes start to glaze over in the first 5 seconds, you realize that our job is to take this incredible science that Dr. Schultz just elucidated and translate it into clinical practice. And so there's the gaining the knowledge, gaining the education, and that was one of the things the expert panel, one of the main points that we came across was that education of the clinicians of what biofilm is critical because it's invisible to the human eye. So how do you explain to a patient that there's something there that no one can see? Even though some clinicians do claim to be able to see it, and these same people usually claim to have seen Bigfoot and have been abducted by aliens, but it's very important that as we move forward with this new knowledge of biofilm, that we have an approach to attack it, because the old school methods of there's an infection or there's a wound, maybe it's infected, maybe it's not, we're going to get a standard culture, we're going to hit it with oral antibiotics, and if that doesn't work, we're going to hit it with IV antibiotics and maybe some topical antimicrobials. It actually gets pretty complicated because over my career I've seen a lot of people fry kidneys with oral antibiotics. I've seen people with all sorts of really adverse reactions with oral and IV antibiotics, and the new antibiotic stewardship rules that are coming out. It's important that we start approaching wounds in a way that treats biofilm, treats potential infection with a topical antimicrobial that is non-cytotoxic and is very effective. So with that said, the science that I get into is the science that is the science that I can really explain to my patients. And what is great about PHMB science is that it's very simple. So what is PHMB? It's a topical antimicrobial. It has the following properties. It's a very broad spectrum, and I'm going to show you the mechanism by which it kills bacteria is extremely broad spectrum. As Dr. Schultz had mentioned, it doesn't rely upon a certain metabolic state of the bacteria within the biofilm. It can kill bacteria in a very broad spectrum method, and well, I'll show you that in a minute. No known microbial resistance. So you have antibiotic resistance genes with many bacteria, and you're also starting to get resistance to silver and other antimicrobials. So you really need something that you don't have to worry about generating resistance or having that resistance there. You also need an antimicrobial that the bacteria just don't tolerate well. And why don't they tolerate it? Because they haven't developed a defense mechanism against it. And sometimes simple is better. High tissue compatibility, PHMB, has been around a long time, and they've done cytotoxicity studies, very compatible with the human body. Very well tolerated by host cells. So here's the mechanism of action. Essentially, you have a negatively charged microbial cell membrane right here. And PHMB as a molecule is positively charged. So when I explain this to my patients, it's something that's very simple they can understand. So the bacterial cell walls are negatively charged. PHMB is positively charged. They essentially attract. And the positively charged PHMB literally punches a hole in the bacterial cell wall, thereby killing it. So here's a picture of that. If you look right here, that's pretty much why I explain to my patients how this works. And it is a very simple mechanism, no known bacterial defense to it at this point. And for a clinician, it's very attractive because when you start thinking about the topical antibiotics and the explosion of the compound topical antibiotics in the last few years, and do you use topical antibiotics, as Dr. Schultz was saying, the study that Dr. Walcott did, where topical antibiotics don't necessarily penetrate a biofilm. So you need a strategy to kill the bacteria within the biofilm and disrupt that cell membrane. It blocks the bacterial attachment, and it helps basically prevent the biofilm reformation. So when we were in Miami, when we all got together, we all agreed that many practitioners out across the country are just not sufficiently aware of PHMB and its advantages. I mean, it's been around a while, and it's been in some gauze products and so forth, but it hasn't really become mainstream the way that silver has. And so in discussing it, we realized that there's a lot of education that needs to happen about your different options for a topical antimicrobial and why PHMB is perfectly suited for wound medicine and how we can treat these wounds and start to address the biofilm. So we also agreed that it's important for you all to understand the mechanism, and I just explained it. Positive charge, attacking a negative charge, punching a hole in the bacterial cell wall, and killing the bacteria. Very, very simple, and my dad, who was actually really a rocket scientist, would say, the simplest answers are always the best answers. And this isn't rocket science. It's a very simple mechanism of how it kills bacteria and prevents reformation of biofilm. And what's really, I think, the take-home message from a PHMB point of view is PHMB kills metabolically inactive microbes, which is what biofilm is made up of, and which is why the topical antibiotics often don't kill the biofilm bacteria, because the biofilm bacteria are metabolically inactive. So that's a really simple and elegant way for an antimicrobial to kill bacteria, especially the metabolically inactive ones. So the next step in sort of creating a solution on how do we approach biofilm-based wound management is pairing PHMB, which really is a very good antimicrobial, with another delivery mechanism that also has efficacy within a wound, and that combination in a collagen matrix with PHMB is a native collagen matrix. So you all are most likely familiar with collagen, but it's important to understand the difference between a native collagen matrix, which is essentially the collagen matrix is in its structure, in its scaffold, is the way it exists in living tissues, versus reconstituted collagen, which is essentially pieces of collagen literally put in a blender, and then the pieces are reconstituted into a sheet. So if you look at native collagen under a microscope, this is what you see. You see this scaffold here that is how collagen matrices are existing in life, in tissues. So the advantages of a native collagen matrix is that you inhibit a broader range of MMPs. Those are those proteases that keep those wounds in the inflammatory state, and they're constantly breaking down collagen and preventing healing, keeping the wound in the inflammatory stage of wound healing. So in Florida, when we met with the panel of experts in literature review, we basically restated what is well known in the literature, is that collagen can be used to sequester proteolytic enzymes. If it may not be effective if bioburden is adequately, or I should say inadequately controlled. And there's some evidence that collagen with native structure inhibits a wider range of MMPs. So that's just when you're thinking about when you're using collagen, and I'm sure everybody in this room uses collagen, and you think about, okay, there's the collagen that you can put on there, that is the reconstituted collagen, and maybe you're not getting results. And you do it week after week after week. And one thought process is you may need to be using a native collagen structure. And that usually comes from skin substitutes or collagen that has been collected in a way that is not just reconstituted. If you do not have adequate bioburden control, collagen products will be less effective in supporting wound healing. So everything that I'm saying, and we're going to get into the clinical, I think, in the next couple slides, everything I'm saying is based upon the idea that if you do not remove slough, necrotic tissue, everything that is keeping that wound in the inflammatory phase, it doesn't matter what you put on the wound. You can do topical antibiotics, you can do PHMB, you can put pretty much anything, any product on the market out there. You're not going to get the result you're looking for, which is to move the patient from the inflammatory phase into the proliferative phase of wound healing. So step number one, you have to debride the wound. You have to remove necrotic tissue. Otherwise, all these therapies will not work. So PHMB within a collagen matrix. So this is really a very unique and novel approach where essentially you take a native collagen structure, porcine-derived collagen, and then you impregnate it with PHMB, and you impregnate it all the way through the graft. And this is actually a bilayer here, and you can see right here and right here, these layers are cross-linked, and then the PHMB is infused all the way through the collagen matrix. All the cells and non-collagen materials are obviously removed, and essentially the reason why you benefit from a two-layered collagen matrix as opposed to a one layer is that it doesn't break down as quickly when it's assaulted by those MMPs in the wound bed, so it resists degradation. Collagen matrix can be fenestrated, it can be solid. Fenestration just allows drainage to come through the dressing, and the collagen matrix is supporting wound healing by providing a substrate for those MMPs, as well as when you combine the PHMB, you're now combining an antimicrobial component as well. So clinically, everything I'm telling you is theoretical, but as a clinician, I have to prove everything to myself. I always approach every product as if it doesn't work, and then I force that product to prove me wrong. And so when I first I got my first sample of collagen matrix with PHMB, which, by the way, as this slide shows, it was indicated head to toe essentially, which was a positive from a rubber meets the road point of view. But when I first saw it, I was very skeptical because I didn't know a lot about PHMB. I knew that native collagen matrix was a solid approach to wound healing. Especially in the chronic non-healing wounds that seem to have stalled, but it really is something that I had to really try for myself. And so I read all the literature. I sort of had an understanding of what it was doing theoretically, and then I actually used it myself, and I actually have a case study here that I'm going to go over in a minute. So once you have this collagen matrix with the PHMB infused, what you want to have is a sustained delivery. In other words, you don't want to put a product into a wound bed and it dumps all of the PHMB out all at once, right? So you want to have something that can sort of the sustained release action, so to speak. So one of the studies here that was done, and this was done actually a year ago in Atlanta, was presented in an animal model, a collagen matrix with PHMB over the course of 72 hours compared to a 2-layer nanosilver, 1-layer nanosilver, basically silver products. And so essentially what you end up with is against MRSA, over a 72-hour period, you have a log reduction that is significant compared to the silver products. Now, why is that? I'm not sure that I'm not sure the exact reason for that, but this is very encouraging because as an industry, we've all used silver for ever since I can remember, for the last 15 years or so, and that has been the go-to antimicrobial topically. And so with PHMB, and when this study came out, it really made me think, okay, PHMB is a pretty effective antimicrobial against MRSA, and again, this was all just data. This is all just literature, and I'm a clinician, so I needed to apply that on the clinical side. So what I did, being as skeptical as I am, is I went to my micro lab, and I took a piece of collagen matrix, a 1x1 centimeter piece of collagen matrix with PHMB, and I plated it on an agar dish. And I did this several times. I did an agar diffusion test. I did 200 microliters of saline. I instilled that into the center of the collagen matrix. And then as the saline leached out of the collagen matrix, it formed a sort of a little moat, I guess, around the collagen matrix. And I incubated it for 24 hours at 37 degrees Celsius. And so the results, and this is what it looked like. So this is the PHMB infused collagen matrix. And this little rim right here is the saline that leached out of the collagen matrix. So I saw that when I actually instilled the 200 microliters of saline. So it was interesting to me that it formed this square perfectly around the graft as opposed to a circle. And so 24 hours I came back, and this is what I found, that there was a kill zone right around the collagen matrix with PHMB. And so I said, okay, well, if it killed the MRSA around the graft, I wonder what's under the graft, right? It was interesting to me because I knew that the PHMB was infused throughout the graft. And so when I lifted up the graft, this is what I got. So this was really a visual for me. I'm a very visual person because when I see a wound, I want to see, is the wound getting better or is it getting worse? And then all the science is kind of going in the background as far as what's going on, but I have to clinically act in a way that heals the wound. So this is what I found. And so I was very impressed with how PHMB as an antimicrobial was able to kill MRSA and create this zone of inhibition. So now that my science side had been satisfied, I really thought, okay, well, I'm going to give this a shot. All right, I'm still going to doubt that it's going to work, but I'm going to give it a try. So essentially, and this actually slide is probably out of order, but the advantage that after we all had used the PHMB infused collagen matrix was that it essentially was a combination product, which combination products are very beneficial because you can accomplish more than one thing at once. So as I mentioned, the collagen matrix plus the PHMB is a sort of 1-2 punch. And what PHMB is doing between debridements is really important. What Dr. Schultz had mentioned is that within 24 hours of a debridement, the biofilm is 1/100th of what it was before. And then after 24 hours, it starts to reaccumulate, and then after 3 days, it's back full force. So as a clinician, I'm thinking to myself, so does that mean I should be debriding wounds every 3 days? Will CMS approve? Will the patients come back? I mean, is that clinically the right thing to do? Wound centers across the country have established just a protocol, and I think it's just out of habit. How often do the patients come back? Once a week. How often do they get debrided? Usually once a week. And then you'll have some people in the community say, oh, well, those doctors inappropriately debride wounds because there wasn't any necrotic tissue. But those people aren't thinking about biofilm because the biofilm is recurring. So now you're talking about something that if the community isn't educated, it seems like you're being over-aggressive in debridement. But the point is that you're really, realistically, in the real world, you're not going to bring somebody back 2-3 times a week to debride them. Okay? Usually their insurance won't even approve. You're lucky to get the once-a-week approval. So now you have to have a bridge between you debride the patient, you remove as much of that biofilm as you can, and then by the time they come back in 7 days, you need something to fill the gap or to create that bridge. So that's where a collagen matrix with PHMB can really fit into the algorithm because essentially you have something that can prevent reformation of biofilm. So here's a case study, and we can get into the real world. The algorithm essentially goes, you have a chronic wound, it's stalled, it's not healing. You might have had repeated rounds of oral antibiotics. You suspect biofilm, which is Dr. Schultz said, suspect biofilm on every wound. And so you've done everything you can to reduce the biofilm. You're still not getting wound contraction. You're still not getting healing. So the treatment strategy is to initially debride the wounds aggressively, but to realize that debridement alone is not sufficient. And then remember that biofilm is reforming within 24 hours of that debridement. And then to use topical antimicrobials. And in this case, PHMB was the choice that I made. Sharp debridement, some people use scalpels. I use curettes. Curettes, in my opinion, you can tear down the layers of necrotic tissue, and you can do it in a very fine motor controlled way that very rapidly you can remove necrotic tissue, get to bleeding tissue. And of course, there is literature that supports that the more often you debride a wound, the faster it heals. And theoretically, that's because you're removing that biofilm. So here's just a classic example. This is a patient of mine that I debrided. That right there is not necessarily biofilm. That's slough. That's just dead necrotic tissue. And so with a good debridement, with a sharp curette, I get a clean wound. So the question is that wound right after debridement, is it clean? Is the biofilm gone? 24 hours, if it looks just like that, is the biofilm there or is it not? I'm not going to bring the patient. How often do you debride a patient, unless you work in a post-acute setting, and you see the wound the very next day? Well, I actually work in many post-acute settings, and so I do see the wounds the very next day. And it's almost disappointing because you debride the wound, and it's nice bleeding tissue, and everybody, oh, that was such a good debridement, and now the healing can begin. And you look at it the next day, and it kind of just doesn't look as good. Maybe there's this dullness to it, or it just doesn't have that exciting, bright future look to it. So when you look at this wound right here, you say, okay, well, it's in danger of recurrent biofilm. And within 3 days, full-blown biofilm. So there's a couple ways. We talked about sharp debridement. One of the things that Dr. Schultz had mentioned was the way that they analyze biofilm is ultrasonic dispersion. And so one of the things that I do in all of my locations is we have an ultrasonic debrider. And I realize that they can be expensive and not everybody has access to one. But what I've noticed over the years is that the wounds that are ultrasonically debrided, they stay cleaner longer. The effectiveness of my advanced therapies is better, and I believe that is because I'm dispersing that biofilm. And so the way ultrasonic debridement works, for those of you unfamiliar with it, there's high-frequency ultrasonic debridement, there's low-frequency ultrasonic debridement. The low-frequency ultrasonic debridement with the contact of the titanium probe, that is going to be the most aggressive debridement you can get. The cells essentially absorb the mechanical energy and from the ultrasonic wave, and the bacteria essentially vibrate and they rupture. So with PHMB, how are we killing the bacteria? We're rupturing them. Ultrasonic debridement, how are we killing them? We're rupturing. So it removes only non-viable tissue, so you can run an ultrasonic debrider over your arm. In fact, before I debride my patients, I pass it over their hand to show them that it doesn't harm healthy tissue. It doesn't leave a mark. It is very benign. But when it comes across necrotic tissue or non-viable tissue, it blows it away. So here's an example of an ultrasonic debridement. This is a venous ulcer, back of the calf, and this is 20 minutes before, 20 minutes after. So it goes from a wound that is smack dab in the middle of the inflammatory phase, necrotic tissue, slough, biofilm, all in here, and now this is a microscopically clean wound. And so it's ready for the advanced therapies. But again, what am I going to do to prevent the biofilm from returning? And just as a reminder, no debridement, no progress. If you look at the dates on this patient right here, and I say no sharp debridement, no progress. This patient actually was getting a topical debrider and was admitted to the hospital July 26th, discharged on August 23rd, and had very little progress. And so I think by now we are all aware that if you just leave necrotic tissue on a wound, it's not going to progress. So the treatment strategy, be aware that biofilm is virtually on all wounds. Just approach the wound, biofilm is there. Treat all wounds assuming that the biofilm is present even when it looks good. And the case study I'm going to eventually get to, which should be the next slide I think, is going to be a good example of that. And then if you focus on controlling your bioburden as the first step, and you're thinking about it from the beginning, you're going to prevent yourself from getting caught in that the wound looks good every week. It looks good every week, but it's not closing. And those are the wounds that usually have the red granulation tissue, and the granulation tissue is almost all the way to the surface, but yet why are we not getting closure? So let's get to our case study. So this is a 51-year-old diabetic male. He'd already had a right below the knee amputation. He was walking with a right leg prosthetic, which means he was putting more weight on his left leg. And he had a Wagner grade 1 diabetic foot ulcer for the last 6 months. He had no osteomyelitis. But you notice he had all this maceration, all this drainage, and his wound culture had showed MRSA. So he'd been on various courses of oral Bactrim, and yet he was still having this drainage, and this wound bed just looked like it was not going to get out of the inflammatory phase. So first step was to do sharp surgical debridement with a dermal curette. And again, it is, whichever instrument you choose, you need to make sure that you're doing an appropriate debridement. One of the things that I see clinically across the country is that people are timid in their debridements, and they don't want to remove that necrotic tissue, and then they do a staged procedure. Let's get the top layer off, and the next week we'll get the next layer off, next one, and let's get it all off now, okay? Be done with it, okay? That's how we're going to start moving into the proliferative phase of wound healing. And then the next step was to offload. So we had to do total contact casting 3 times a week, which is not common for us. We usually do it 1 time a week. Why do we have to do it 3 times a week? The drainage was so bad, and we'd even tried a negative pressure in the vac, and he macerated with that. The drainage was so bad, and I thought to myself, okay, we've given him oral Bactrim, we've offloaded them, we've debrided them, and we're still getting all this drainage. So in Louisiana, we use a lot of gentian violet, and I suspect that tradition is because it's purple, and LSU's colors are purple and gold. But you'll see, whenever you see pictures from Louisiana of wounds, if you see that purple around the wound, what does it mean? It means there's maceration, and someone is trying to treat maceration. So with all the efforts that we just gave, to try to heal this wound, this is what we ended up with. It looks pretty good, right? Red, granulation tissue. We still had the maceration, though. Had to change that cast 3 times a week. And so my nurse manager said, look, Dr. Carpenter, we need to do something with this because we can't see this guy 3 times a week. The insurance company's getting angry. And so I said, okay, well, this is right about the time that I came across the Collagen Matrix with PHMB, and I'd done the microbiology study. I'd done a little bit of research and just really decided, okay, well, I'm going to give it a chance, but I'm pretty sure it's going to fail. So continued drainage and failure to heal despite debridement and casting. The idea here was maybe I could prevent biofilm reformation and get this wound fully out of the inflammatory phase using the Collagen Matrix with PHMB. So my application strategy was to debride the wound, get it nice and bleeding, and from my experience with PRP and other collagen products, I always like to soak it with blood. It's the patient's blood, their growth factors. It's not going to hurt the patient, and I can't tell you that this is supported by literature, that this is the way to do it. It's just the way I do it. Plus, I think it looks kind of cool with the blood soaked into the graft. So I applied this, and then the patient came back, and we kept it on there for 2 weeks, and this is what we ended up with. So what was really cool, and the size difference was important. I mean, it decreased in size. But what's really cool is there's not a lot of that gentian violet, right? Look how dry that peri wound is. So we were able to go from 3 times a week casting down to 1 time a week casting. So that's what really got my attention, because that was such a problem, the drainage, and there wasn't anything else that we did different. He was getting casted, we were offloading, we were debriding, and so I thought to myself, okay, something happened here. Did we prevent reformation of biofilm? I don't know, but it certainly showed a decrease in the inflammatory, as Dr. Schultz was saying. There's the infected side and there's the inflammatory side. It just showed a decrease in inflammation. And so we reapplied it weekly for 6 weeks, and here's what we ended up with healed. So after this, I said, okay, well, I'm going to give this product a chance, because it's starting to prove to me that it's doing something that the standard of care wasn't doing. And so that's when I really started to research more and get involved more with PHMB infused into a collagen matrix, because I saw a result that because it's a head-to-toe product, there are a lot of patients, there's pressure ulcers that I treat that are stuck in that inflammatory phase. Surgical wounds, there's so many wounds that get stuck in the inflammatory phase, and having a head-to-toe product was really important to me, so that clinically I could start to approach a biofilm-based wound management strategy with something that was going to directly address it, as opposed to just hoping that my oral antibiotics or my topical antibiotics would do the job. And so there's the transition, and that's a really good transition. You can see the drainage decrease dramatically, and patient did very well. So a summary of the panel, we basically came up with the collagen matrix with PHMB provides a two-pronged approach with collagen and antimicrobial. And we were looking at DFUs and VLUs, but really with all wounds, there's an approach that if you can use a collagen matrix with PHMB initially and get it out of that inflammatory phase, then you can transition to your cell-based strategies. And obviously, you don't want to advance to the cell-based strategies until you've gotten it out of the inflammatory phase. And debride, control bioburden, and in combination with collagen matrix and PHMB, it was a strategy that worked, and it's something that we're exploring more. So, any questions?
PHMB, but you talk a lot about the cytotoxicity of silver. Is there, and all the silver dressings have different parts per million, is
Yes.
there a known parts per million under which it's effective but not cytotoxic, and over which it's cytotoxic? Do we know?
For silver?
Yeah.
That's a great question. I'm going to get a little technical with you. It depends on the form of the silver, whether it's ionic silver or it's nanocrystalline silver or if it's superoxidized states of silver. So those all have different cytotoxicities because they interact with the cell components in a different way. There are some published literature that talk about the different levels of cytotoxicity. I'm going to be general. Ionic silvers tend to be more cytotoxic than non-ionic silver. See how generic I'm being here? And the superoxidized silvers are the newest phase of this, and the literature is contradictory, in my opinion. Some literature says they're less cytotoxic than nanocrystalline silvers. Other reports say they're even more cytotoxic than silver ionic crystal Ag+. So I think what you can hopefully rely on is that for any dressing to be approved by the FDA, it has to have one of three claims. And remember, no microbicidal dressings have the claim of reducing the level of bacteria in the wound. That's a drug claim. So these are devices. And the three claims the FDA allows in packaging is it prevents bacteria penetrating through the dressing to the wound surface. That's the bacterial barrier claim. It prevents growth of bacteria in the dressing, not in the wound bed. And it preserves the dressing while it's in storage. So it's the shelf life extender, okay? Notice none of those say anything about what it does on the wound bed. But they also all have to pass minimum cytotoxicity levels, which means that in general, you could assume that if used according to the instructions for use, that the cytotoxicity would be acceptable if you use it the way they say. Now, there are reports in the literature of patients developing argynuria, which is silver toxicity, due to overload of silver from dressings penetrating into the blood and causing systemic problems. What kind of patients are those? Those are usually pediatric burn patients with greater than 60% total body surface area burn, so they're getting silver almost all over their body, right? That's not a diabetic foot wound. So I'm trying to put it into some perspective as best as I can understand and explain it. So there are also credible reports that show in animal models where there are partial thickness donor sites without any bacteria problem that silver dressings applied to that will retard the rate of epithelial healing. Okay. So at the University of Florida, the level 1 burn trauma unit, my great colleague Dr. David Mazingo, treats patients that have huge burns. They are at high risk of developing infections and systemic infections that will kill them. So in many of those patients, they make the clinical decision, we'll take a hit on some of the wound epithelialization to prevent the risk of a systemic deadly infection. So what does that mean? You have to balance how much risk a patient is under if you use a microbicidal dressing that has been reported under certain conditions to impair healing. But I think in general, you can assume that silver releasing dressings used under the instructions for use will not totally kill your wound cells. Now, I'm going to also say that wound irrigation solutions also have to pass safety and cytotoxicity. We had a clinic at the VA in Lake City, which is just north of Gainesville. And my wonderful colleague, Linda Cowan, Dr. Cowan, and I went up and I gave the biofilm talk. And we talked about biofilm-based wound care, using good microbicidal treatments after debridement. Thought we had it all clear. We came back in 1 month to follow up. Staff brought out a patient and said, look at this great wound that we have treated with your biofilm-based wound care. We've irrigated this patient 3 times a day with dilute bacons. There was no biofilm on that wound. There was also no living cell in that wound. So you have to make the clinical decision about how much of a hit will you take on killing wound cells if you have a high probability that you still have some biofilm left. The concept of step down then step up therapy says use your most effective therapies first to remove the biofilm and planktonic bacteria, because that's what's causing the inflammation. When that gets reduced down, now you have the option of moving to another dressing, one that may have less cytotoxicity but may not be as effective as your biggest gun at killing biofilms. And that's when you have to make this clinical decision. And then finally, when is that wound bed prepared well enough that you can use an advanced therapy like a collagen dressing and jumpstart that wound and help it heal some more? But as Dr. Carpenter said, one of the things that disappoints me the most is when I see my clinical colleagues who are great clinicians have a patient comes in, 6 months of a non-healing wound, they debride it and immediately put on an $1,100 amniotic dressing. 3 days later, it's gone. And that's because the wound hasn't had time to reduce the proteases and inflammation in that rapid 3-day period. I mean, proteases don't just disappear. They debrided, but the biofilm-induced inflammation, the neutrophils that remained within the wound, were still high enough that the level of the proteases were so high that it just digested that amniotic membrane. Doesn't mean amniotic membranes aren't effective. They are terrifically effective when the wound bed is prepared well enough and that advanced therapy is ready now to step up to that kind of a very effective treatment. So that's a long discourse. To answer your question, is there cytotoxicity? Hell yeah, there's cytotoxicity to every microbicide, but it has to be balanced.
How often do you have a culture that's not really getting that bioburden versus another culture that, how often do you miss the bioburden?
Well, are you talking about biofilm? Okay, biofilm. You want to answer that question?
So that's a really important question. Remember, let me just remind you that the typical clinical microbiology laboratory that you'll send your sample to does a real great job at culturing planktonic bacteria that can grow on agar plates. They do not disperse the samples adequately enough to take the bacteria that are in a biofilm in a non-culturable state and expand it out, break the quorum sensing so those bugs will become metabolically active and will grow again. So it doesn't mean that the clinical microbiology laboratory data isn't useful. It is. We have 150 years of evidence that certain levels of planktonic bacteria are going to impair healing. What we now know, though, is that if you use DNA sequencing to identify bacteria species versus standard culturing techniques to identify bacteria, the culturing detects 17% of the bacteria species on average that's in a wound. They almost never detect the strict anaerobic bacteria, which can be a major component, especially during the early stages of treating a chronic wound. So if I understand your question, how frequently and how dispersed within the wound bed would a biofilm potentially be? In other words, how consistent is the sampling, or are you missing a lot of the planktonic bacteria and biofilm bacteria? Dr. Thomas Bjornholt at Copenhagen University has a great paper published on this. He took a series of patients and took punch biopsies at the 4 o'clock hours, 12, 3, 6, and 9, and in the center. And he used biofilm dispersing techniques, so he's getting all bacteria. It turned out that there was tremendous variation within the CFUs that were detected at the edge and also in the center. And one case particular, I remember, he had 8,200 CFUs at the number 3 clock hour position. When he went directly across the wound to the 9 o'clock position, he had 50. So that shows you the heterogeneity of the bioburden, including biofilm, that's within a wound bed. They are not uniformly distributed as a single layer across wounds. They're very heterogeneous in their distribution. Also, when he looked at the types of bacteria that were identified at these 5 selected points, there were bacteria that were common among all those, but there were also some very different patterns depending on what part of the wound you sampled. So the take-home simplified message is sampling makes a huge difference in what bacteria you're going to detect and how much bacteria you're going to detect. And standard clinical microbiology labs do not differentiate or detect biofilm. They detect culturable planktonic bacteria, and on average it's about 17% of all the bacteria species that we can identify with DNA sequencing. Doesn't mean those data aren't useful. We have 150 years of data that says there is some information that's important in that. But it also means that it's a very incomplete sampling. And so you may design an antibiotic, oral or systemic or topical antibiotic treatment that targets these bugs, because that's what the clinical microbiology lab said was there. And Dr. Carpenter will talk even more about this too. But when you look at the DNA analysis, you may be missing some major pathogens.
And I'll just add to that, and just make sure you're always getting a deep tissue culture. Some people, the DNA culture labs are saying you can do a swab, and just ignore that. Just make sure you're getting deep tissue. So clinically, if you want to do 2 cultures, and that might help you as you develop your own protocol, start getting 2 cultures and start comparing them from 2 different parts of the wound. I don't know if the lab will appreciate 5 in one wound. I think are we out of time? So we're getting the hook. But if you guys would like to have some questions, feel free to come up here and we'll answer them. All right.